All right. Good Friday, everyone. My name is Gregory Saunier. I will be introducing Jason today for his talk. It's a real pleasure to introduce him. Thank you very much, Jason, for all your help you provided at the ranch, whether it's like data collection in the field, chemistry lab, or ranch lab work. We're really excited about this presentation because Jason is the first person to really look at one group that we have not looked at on the ranch, which are zooplankton. So we're really excited about this talk, and I hope you are too. A few words about Jason. So Jason is very passionate about zooplankton, and you'll probably see this from that presentation. And if you really want to rile him up, you just mix up ostracod and rotifer, and you'll get going on it for about 30 minutes. Another passion of Jason is seashell. So if you saw some seashell disappearing on your window, maybe it's him. And finally, something that I discovered today, he has another passion for ice cream, but only when there is no cookies in it. So Jason is originally from New York. You see, I didn't say New Jersey. But he actually studied in Florida at Nova Southeastern University in Fort Lauderdale, where he majored in marine biology. And so he had a really atypical background for an intern we hire at the ranch, but he was really motivated by the work we're doing here and really interested by what's happening at Buck Island Ranch. Before coming to the ranch, Jason had experience working with and identifying zooplankton in a lab in Delaware. So this is where he kind of honed his skill at identifying zooplankton. So that served him pretty well for this internship, I would say. And before finishing, I would like to announce that he has accepted a position as a wildlife technician for University of Florida Ranch Cattle Research and Education Center at ONA, where he will work with Ance Ellington. Thank you, Jason.
Howdy, y'all. Thank you for coming. Thank you for coming. Thank you so much for Greg for introducing me, and thank you for letting me present here. So as you know, my name's Jason, and I'm an agroecology intern at the ranch. And today we're going to be learning about zooplankton variability in Buck Island Ranch's wetlands, micro life, macro impact. So first, we're going to start off with what exactly are zooplankton. So zooplankton can be classified as microscopic drifting organisms. First things first, they're heterotrophs, part of the plankton community, not to be confused with phytoplankton, and they're virtually found in all aquatic habitats, ranging from our drinking water, caves, oceans, and lakes. While they might be tiny, zooplankton are very important for a myriad of reasons. Foremost, they serve as a crucial link from the primary producers of phytoplankton to higher trophic levels, such as other macroinvertebrates and fish. In addition, they help to regulate algal blooms. They're involved in capturing carbon, nutrient cycling, and they serve as bioindicators. Shifts in their community structure can reveal changes in water quality and a multitude of other factors. So on the ranch, there has been a lot of previous research on how bovines affect macroinvertebrates, but there has been no research on how their grazing presence or otherwise of cows affect one of the most important groups of invertebrates, zooplankton. Additionally, there has been a lot of zooplankton research in the state of Florida, but unfortunately, it's mostly focused on either estuarine zooplankton or zooplankton in Lake Okeechobee. So this project, it was created in order to address these gaps in knowledge and help us learn more about zooplankton. The main goals for this project were characterizing the zooplankton biodiversity on the ranch, helping to get our INAT numbers up. Additionally, examining the effects of wetland fencing upon the zooplankton community. I hypothesized that grazing exclusion would lead to a greater zooplankton species richness and better water quality. In addition, also, that the improved pastures would have a lower species richness compared to the semi-native pastures. due to reduced nutrient loading, lower disturbance effects, and greater habitat heterogeneity. And finally, to assess zooplankton species as potential bioindicators of wetland health. Basically, for this, my hypothesis is just that the populations and abundances of rotifers and cladocerans specifically would help reveal the conditions of the wetlands. So for the study area, I know you're all familiar with Buck Island Ranch, and there's two types of pastures. The green ones are the semi-native, and the reds are improved. The improved are managed more intensely, and there is legacy fertilizer residue in them. And a total of 16 sites were sampled throughout the ranch, some in South Marsh, East Marsh, and West Marsh. And of course, as I said, I want to include wetlands in semi-native over here and improved and use wetlands that are both fenced and open to capture all of the conditions. Finally, for spatial collection, between the dates of November 22nd and December 17th, all 16 wetlands throughout the ranch were sampled. Typically, a plankton net is used to capture plankton, but on the ranch, since the wetlands are very shallow, I used bottle samples instead to combat the issue of plankton patchiness and to get a representative sample of the entire wetlands. I used 3 1-meter transects in every wetland and collected 1 liter of water sample for nutrient analysis at the UF Analytical Research Lab for total nitrogen and phosphorus, and in addition, readings from the YSI and turbidity meter. For the temporal component of my project, I chose wetland 188 right here, since it looked very productive and it had one of the deepest water depths, so I knew that it would last well into the dry season for my sampling. So every 3 weeks on these dates, I collected samples and then observed them underneath the microscope. Once back in the lab, the water was filtered through a 250 micron sieve to remove all detritus and larger macroinvertebrates, and then it was passed through a 38 micron sieve in order to retain the zooplankton. This concentrated sample was then poured into a sample bottle and preserved with Lugol solution in order to help preserve and stain the zooplankton to make them easier to see. And I know some of you are probably questioning, how can you identify zooplankton when they're so small? Well, as you see here, there's a multitude of different features, and I consulted local taxonomic guides and research papers to make sure that my species identification was correct. Papers on zooplankton in Lake Okeechobee and Lake Apalaka helped significantly with this. And here I just labeled some of the most basic features of Daphnia and a copepod found. These two were actually found in Lake Annie. And for my analysis, I did a bunch of different R biodiversity indices, box plots, NDNS, two-way ANOVA, and CCA. Now for the species, the most interesting part, a total of 37 different species of zooplankton were found representing 4 major taxa. First, we have ostracods. They're a microcrustacean. Basically, you can imagine them as a small shrimp shoved into an almond shell. Oftentimes, right here, you see its little antenna pop up to feel food and find other resources. Then we have copepods. If any of you have watched SpongeBob before, this is plankton. They're pretty abundant. In fact, they're the most numerous multicellular animal on Earth, and they're most commonly found in the oceans. And 3 species and 1 larval form was found here in the waters of Buck Island Ranch. And my favorite group are cladocerans. They're filter feeders. They can be identified due to their singular eye, carapace, and filtering appendages. They're also called water fleas since they bounce around like little fleas in the water. And lastly, we have rotifers, one of the most unique groups of animals. They're very basic pseudocoelomates, and 22 species were found. And zooplankton have a variety of adaptations in order to survive in the changing conditions of Buck Island Ranch's wetlands. Now for results, here we have a species curve. So I counted over 7,000 individuals of zooplankton while I was here, and we see that it is mostly dominated by copepods and rotifers. Ostracods, the single species, is low in the curve, and the cladocerans are much less abundant. And here we have this very cool graph. And basically, this box plot illustrates the effects of pasture type, improved versus semi-native, and grazed versus fenced on several different water quality parameters. And the ones that were found to be significant are total P, total kids nitrogen, turbidity, and DO. It was found that significant interactions between pasture type and grazing treatment were observed for these. And basically, it just indicates that pasture condition, either improved or semi-native, may influence nutrient and oxygen dynamics independent of grazing. And for the spatial component here, we have wetland 127. Now, this was the most diverse wetland due to its high Shannon index and other biodiversity indices. What is very interesting is that it's in an improved pasture and it's fenced. There was a ton of Daphnia ambigua, which thrive in good conditions. And here was the worst wetland. Wetland 310, it had the lowest biodiversity index numbers, and it was dominated by rotifers. This species here, Crotalus serrata, is a bioindicator for bad water quality. And then here, we have a box plot of species richness compared to improved and semi-native wetlands along with fenced and grazed wetlands. And it was found that there was no effect of pasture type or grazing pressure upon zooplankton species richness, which was very interesting. This can be due to the fact that there was only 16 samples and there needs to be done, or either that it needs to be a temporal study in order to study the shifting communities longer. In addition, there was no effect of pasture type or grazing exclusion upon Shannon diversity. And here we'll move on to the temporal study for wetland 188. You can see here in the wet season and dry season how drastic the conditions are. So plotted here is zooplankton composition and water depth over time. And we have the four major taxonomic groups. From November to January 9th, it was a pretty heterogeneous community, mostly dominated by rotifers. But then on January 30th, as the water level lowered significantly, it started to be dominated by copepods and cladocerians. And then February 18th, when the water level was only 21 centimeters, it was almost fully dominated by copepods, and we had a guest appearance from ostracods. The reason why I think the ostracods appeared is that they're mostly a benthic zooplankton species. So since the water level lowered, it's possible that they were more abundant in the water column, or the conditions just favored them better. And additionally here, we have the same dates, but zooplankton feeding strategies over time. Zooplankton have many different feeding types, but I just minimized it into these three main groups. We mostly have a somewhat balance between filter feeders and omnivores and predators, which is very indicative of a healthy wetland. But then on the dates of January 30th, February 18th, we see a shift to more detritivore-based food web. And this shows that it's really unhealthy because a lot of detritivores mean that the ecosystem is changing and there's too much detritus overloading it. Then here we have the decrease in Shannon diversity over time. And you can see here as time progressed, the Shannon diversity decreased. On these last two dates, it was dominated by copepods, rotifers, and ostracods. And there was an influx of 6 new species, which were previously not seen in any other wetland. And all of them were the detritivores, which is very interesting. And here we have cladocerians and rotifers as bioindicators of eutrophication. And with this NDS, we can see that zooplankton community composition differs with pasture type and nutrient conditions. Improved wetlands are mostly clustered over here near conditions such as total P, total N, and turbidity vectors, while semi-native wetlands are clustered near temperature and DO. Eutrophic indicators are Maxithrix rosea and Ceridifnia. These are two species of cladocerians, and Curatella serrata, which is a rotifer. These are indicators of bad water quality and eutrophication. So basically, if you get a wetland sample of zooplankton and there's a lot of them in there, they're just indicative of eutrophic conditions. And this is true, supported by the literature of zooplankton in Lake Okeechobee. While over here, we have more oligotrophic stable indicators such as Daphnia ambigua, Arctodiponomus, and Brachyrium, which are other cladoceran species. And they're found in more semi-native, less impacted wetlands, also supported by literature of zooplankton from Lake Okeechobee. So for discussion, we have found that 37 species of zooplankton were identified on the ranch, which is a lot more than I expected. Usually zooplankton are less diverse in wetlands, especially in mid-latitudes like Florida. In addition, there were some species that were really rare and were not found elsewhere, such as in Lake Okeechobee or Lake Appalachia. Secondly, we found that the exclusion of cattle had no effect upon zooplankton. Location in semi-native versus improved pastures had no major impact upon zooplankton, but it did on water quality. And 4, we designated zooplankton species as bioindicators on the ranch. For future directions for this project, a long-term temporal study for multiple wetlands would be very effective to see how the changing conditions impact zooplankton and the appearance of new species. Use eDNA to confirm identification and cryptic species. While I am very confident in my species identification, you can never be completely sure. So I feel like this would be very good to confirm it. The collection of phytoplankton for more analysis. If you collect chlorophyll A, you could do a whole bunch of wetland index formulas to truly see how healthy a wetland is. And finally, measurement and identifying the sex of zooplankton. I tried my hand at measuring zooplankton, and if you measure them, then you could get their biomass, but I felt like this wasn't really accurate since I had a small sample size. And by identifying the sex of zooplankton, or if they're either sexually reproducing or through parthenogenesis, you could find out if the wetland is healthy or not. Special thank you to Betsy, Greg, everyone else on the ranch. And here we just have some cool pictures of zooplankton. Some nice pictures of phytoplankton found on the ranch. And additionally, we have a whole bunch of worms, gastrotriches, which is really cool. There was a few tardigrades, flatworms, and other ciliates found. Protozoans are another type of zooplankton used in biodiversity indexes, but I had never identified protozoans before, so I just exclude them for the study. But now I feel pretty confident in identifying, and going forward, they should definitely be included in zooplankton counts on the ranch. And then here we just have some more cool pictures of little guys. Lahas?
I'm trying to see how I can do this. Can you turn your speaker off? Okay, obviously this is working, so.
Okay, great. Thank you, Jason. That was a really cool talk, like also really great photos. What is do any of these ponds completely dry out? And is what species found there driven by which species can persist as eggs in the soil?
I'm glad you asked. I made a slide just for that. So. Turn on your audio. Yeah. Okay.
No, I was saying turn off your speaker.
I can't.
Okay.
I'm sorry. It's not working. So literally all species are able. Sorry.
Yeah, yours, I think you're still muted.
Okay, so zooplankton species, all 4 taxa have really unique adaptations such as diapause eggs. So when conditions improve, their eggs can spawn. This is a cladoceran egg. It has a thick layer of ephema around it, so it's able to resist all types of different conditions such as high levels of UV, drought, anything else. So when conditions are more favorable, then it spawns. They can also do cryptobiosis. Many rotifers do that, and in addition to all the other worms and gastrotrichies found. Does that answer your question, kind of like? Yeah. Yeah.
Thank you. Tremendous amount of work on the microscopy. Well done. And you concluded that there was no impact of the cattle on the Shannon diversity, the species composition. Yes, I think that's the one I wanted to look at. Bingo.
Yes. No movement.
And so it's actually interesting to me, like I think there's actually maybe something going on here on at least on the improved pastures and maybe both sides. I can understand how the statistical tests maybe show you there's no difference in the mean, but you get that huge variability. So what do you think that might mean, especially in the improved pastures?
I feel like it's more so a lot of the wetlands I visited in improved pastures had greatly different conditions, not only in like nutrient levels, but landscape. The plant communities were completely different in some of the improved pastures. So I feel like sometimes plants are reflected in this. Sorry, plants affect this too. There is previous studies done on how plant communities, especially submerged plants, impact zooplankton diversity and the species present. But I just didn't consider that in this study because I'm not really adept at studying plants, and it would add a whole complex layer.
Yeah, I mean, to me, this actually looks like you do have something really cool here and consistent with other studies of grazing indirect effects on other herbivores or even predators, because you've got potentially very low diversity on the improved pastures when you don't have cattle, but then you add them and they're probably doing something to increase the habitat heterogeneity, and you have higher diversity in all cases when you have the grazing. That's pretty neat, and you're only reaching that diversity in the fenced ones where you have the very highest levels of habitat heterogeneity naturally in the pond maybe. So I think you've actually undersold your results, and it's even cooler than you thought.
For the grays, there was also a very low sample size too, so that's also why it might be like that. But yeah. Any other questions or?
There was one little guy you pointed out in particular at the end that you said was very exciting to see. And I was wondering if you could repeat what that was and why you were so excited to see it.
Oh, just it's called a gastrotriche, or where is it? Right here. It's just like a very simple animal, if you can even call it animal, and I've just never seen it before. I learned about it in one of my classes, and it was just cool seeing it in the wild. I would like to know, I was kind of rushing through my presentation, so I kind of forgot to say this. Another interesting organism I saw was a species of Cladoceran called Daphnia lamuts. It's the African water spiny flea, and it's invasive, and usually it's found in Lake Okeechobee. When I was doing my preliminary testing for all the wetlands, I found a few of them in one sample, but then when I went back to that wetland a week later to see if I could capture any more, they were no longer there, so there is the potential for invasive zooplankton on the ranch. All of them in my study were native, except the one I found that was just in the early part of it. They can get over here due to like aeolian dispersion from Lake Okeechobee through the canals, or who knows if someone brought a boat over into one of the canals and they just happened to get into the wetlands due to flooding or the flooding of the ditches.
Oh, it's a phone
It's
now.
just my phone, yeah.
The phone. Um, the idea that you just brought up of invasive zooplankton makes sense. It's just never something I've thought about. That's really cool. Are there any kind of management practices for invasive zooplankton?
There's no like real management practices solely focusing on zooplankton. Obviously, if you go into a lake and you bring in a boat, you're supposed to clean the bottom of the boat so you don't transfer other invasive species like hydrilla. But for zooplankton, since they have very special adaptations, if some of the eggs get onto the side of the boat, they can last there for months, even years. So if you bring it from Lake Okeechobee, let's say to Lake Placid, and the eggs release into the lake, they can transfer that way. Otherwise, for invasive marine zooplankton, they can transfer from one continent to another based on ballast water. There's a species of crustacean called caprella that's greatly expanding throughout the world due to ballast water. I don't really know how to regulate the releasal dispersal of ballast water for zooplankton. Maybe you can try to have some type of micron filter, but I don't know how effective that is. Another way is they always release the ballast water away from like the continental shelf recently. That's like a study on that, but I don't know how effective that is. But for zooplankton, I think they're just traveling around regardless of any precautions. Because let's say a whistling duck is in Lake Okeechobee with some of the invasive spiny water fleet in them or on them, and they go to one of the wetlands here, it's gonna get out like that. So